Les Recio, the Chief Scientific Officer of ILS, and I'll be giving a brief presentation on ILS capabilities. ILS is headquartered in Research Triangle Park with a focus on toxicology, including computational toxicology, reducing reliance on animals, genetic and molecular toxicology, in vivo tox, pathology, and histology. We have a 3-person quality assurance unit. We do study designs compliant with regulatory agency requirements and OECD test guidelines. We have a state-of-the-art laboratory and animal facility, which includes cell culture, genetic and molecular toxicology, as well as an ALAC accredited animal facility. About 1/3 of our staff hold advanced degrees. We have an extensive peer-reviewed publication. We also have a number of boarded anatomical and clinical pathologists on site. We also participate in international scientific and expert working groups, including the OECD expert working group involved in genetic toxicology. We develop novel methods via research grants, we serve on editorial boards, and we are known to integrate novel approaches of molecular and cellular biology into traditional toxicology. The in vivo toxicology group conducts traditional rodent regulatory toxicology studies, including acute, subchronic, chronic studies, the RAS-H2 mouse, the extended one-generation assay, a reproductive toxicology test, a number of genetic toxicology tests, and endocrine disruptor screening program assays. We do safety assessment with higher species via partnership. We do a mode of action study, which typically are next-gen sequencing expression profiling studies. Dose formulation in-house. We have a number of software systems, including Augmentus, which allows real-time telepathology. We also have a genetic and molecular toxicology group that conducts all the genetic toxicology tests required by regulatory agencies, includes a bacterial mutation assay, flow cytometry, micronucleus assay, chromosome aberrations, in vivo micronucleus, and comet assay. We also do a number of follow-up assays, which include the in vivo PIG-AG mutation assay, the multi-flow assay, which is a newer assay that uses biomarkers to predict clasogen or antigen mode of action, genotoxicity testing in hepatocytes, as well as screening assays in genetic toxicology, which includes computational QSAR to assess for structural alerts. We also have a molecular toxicology facility at ILS. This includes expertise in designing and analyzing gene expression. This includes in the laboratory doing quantitative targeted RT-PCR for specific genes, as well as next generation sequencing or so-called whole transcriptome profiling to get at mode of action. We also have a computational toxicology group, which is all about reducing reliance on animals. This includes analyzing medium and high throughput data to develop in vitro to in vivo extrapolation modeling, cheminformatics to conduct data gap filling and read across. We have a data science group that does bioinformatics, for instance, for next-gen sequencing to identify differentially expressed genes and pathways. And finally, an information science group, which can do toxicology reviews of specific agents and develop high-quality databases. Thank you very much for your time, and please visit us at ILS. We published recently an ILS newsletter, which gives racing data on what ILS is up to. Thank you.